So we'll go ahead and get started. I don't want to penalize you for being late or on time here. So this is the Data-Driven Ag webinar series. I'm Dennis Buckmaster, professor in Ag and Biological Engineering, and also serve our administration and community as a Dean's Fellow for Digital Agriculture. So this webinar series is weekly. Most of you know you've been here week after week, but not necessarily everyone. If you missed previous sessions, they are all recorded and archived. We can go to the next slide. This work is, of course, Purdue sponsored. You can tell from the logo and the color scheme here, two of our favorite colors, black and old gold. But we do have a Digital Ag Resources website that archives all of these, as well as has a very good directory of who's doing digital ag things at Purdue, resources at Purdue and beyond, and also covers our educational programming, formal and informal. There's a specific site regarding UAVs. And as you think about the topic of next week, you may want to go there. And then much of this work is in part supported by the Wabash Heartland Innovation Network, which was a Lilly Endowment Foundation gift to the 10-county region here in West Central Indiana. So next topic is practical applications for UAVs. So we've got faculty and staff from agronomy, electrical and computer engineering, ag economics, and then ag and biological engineering in those next 4 weeks, and we hope that you can come back and join us for those. So during today's webinar, it's going to be a little different than our usual format. Rather than a presentation, we're going to do it a little bit more like a sort of dialogue talk show between three of us here. So we'll get to that in a minute, but we would encourage you to extensively use chat. If you have a question at any time, just open chat, type in your question or your comment. We welcome that. And that will probably nicely complement and influence the direction that this conversation goes. At the end, we'll also allow you to unmute and pose some questions live to our presenters if you'd like. So that covers my introduction. Now we can switch to Mohit, maybe your first slide. Let me just provide introduction. Today we have Dr. Mohit Verma from Ag and Biological Engineering, a friend and colleague here at Purdue, and also Aaron Ault, who is a farmer, computer engineer extraordinaire. We call him our chief data architect in the Open Ag Technology and Systems Center. Both of these guys are good friends and colleagues here. I'm very pleased to have them present this topic today, which is about livestock disease and smarter treatment of those diseases. So I think we'll just go ahead and start with, and you guys can chime in, through our dialogue here, a little bit about your perspective and that sort of thing. But we'll start with Aaron, and maybe you can just go ahead and get rid of the slide there for a second. Aaron, you're a farmer. You have lots of cattle, lots of land, some smart machines, probably some less than smart machines, etc. Tell me about what is this bovine respiratory disease? How does it affect the beef industry here? And how did you meet Mohit?
That's a lot of questions all at once. I'll see if I can tackle them all at the same time. Thanks, Dennis. So yeah, as you mentioned, I am a farmer here. I've got in north central Indiana, about an hour and 15 minutes northeast of Lafayette. And I've got about 3,000 head of beef cattle in confinement facilities and about 3,200 acres of corn and soybeans that we raise as well. And on the beef cattle side, what originally brought Mohit and I together around an idea was me complaining when there was an ag and engineering get-together event trying to cross-pollinate the different departments. And even though I was a farmer, I work in electrical and computer engineering because that was my graduate degree, was in computer engineering. And so the ag and computer engineering got together, and Mohit and I happened to sit beside each other, and it turned out to be very fortuitous. Because I was complaining of that the number one thing I dealt with cows every day was this disease complex called bovine respiratory disease. So it's not just one disease. It's essentially a whole class of diseases: Pasteurella Mannheimia hemolytica, Pasteurella multocida. There's several different ones, and each of those have their own strains within them. that essentially fill a calf's lungs with fluid over time. And so that's why we tend to call them all pneumonia, colloquially. But the hard thing about them is they always have these, some small amounts of some of these bugs around all the time. And when they get under stress, like shipping, or another cow, accidentally rolls over on them wrong or something, or the temperature gets out of whack with how the length of the hair coat is, all of a sudden these things will start to take over and it can kill the calf extremely rapidly. And so I walk through about 1,000 head every day in the morning, looking through them all, trying to find any calves with the very first signs of that they're thin, their head's down a little bit, there's a lot of snot running, stuff like that. Because if you don't and get them in to try to treat them, then they're going to die, sometimes within 24 hours of the first symptoms that you see.
Well, you'd sort of think if this disease has been around for a while, actually, I guess it's bovine respiratory diseases, as you describe it here. If it's been around a while, like, why haven't we solved that? And Mohit, you're not a veterinarian. So how did your involvement in this really get going?
Yeah, so as you heard, Aaron, I was hearing the same story as he was sitting next to me, and he was kind of pointing out that, if only I knew which bacteria was causing this disease or which specific one, I could figure out which treatment is more likely to be effective and work quickly, right? Because a lot of times what happens is the symptoms might be similar, but the causative pathogen is different, and then treatment can be different. And especially with the emergence of antibiotic resistance, a lot of times these antibiotics don't work. And that's what you want to try and figure out. So when I was kind of hearing this, I was like, I was aware of the PCR technologies that are out there, and I was like, well, that should be pretty doable. You should be able to detect which bacteria is present in the calves, but then we looked a little bit deeper, and there wasn't a technology, and that's when we were like, okay, let's try to solve this problem. So we started working on this from there on, trying with the goal of really trying to figure out if you have a sick cow, can you figure out which bacteria or which virus is causing that disease, and therefore guide the treatment, trying to make it more effective, trying to get the response quicker, and also trying to reduce excess use of antibiotics, right? So if it was just a viral infection, then antibiotics aren't really going to do anything. So you probably don't want to give antibiotics. So with all those goals, that's when we started about almost 3 years ago now. And we've been going from there.
Cool.
I want to add to what Mohit just said. Not only is sometimes the symptoms the same, like every disease the cow gets, the symptoms are the same. They all look the same. It doesn't matter what they have. They stop eating and their head's down and they look mopey. And it's like, when you're trying to figure out what to give this cow, you have only 2 pieces of information: what I've given the other ones lately, and the fact that he's sick. And that's it. And so the idea that we could have some new information was really exciting to me, because it would just, it would more than triple the amount of knowledge that we had at the shoot when the calf is there and we're trying to figure out what to treat him with.
Oh, nice. Yep. And Mohit, you're a sensors guy at heart, right?
Yeah.
So had you worked on sensors like this before? Like what's
exactly? So my training actually comes from biosensors. My PhD was on developing these gold nanoparticle-based sensors, where we're trying to see can you detect different types of bacteria and see a color change. Then during my postdoc, I was spending some time at Harvard, where I started learning about how we can make these paper-based devices. And there, really, what we're trying to do is make these tests that are typically done in the lab, but make them simpler. So what you can see on the top left is this little kit, which the right one is a dropper. So it's, you can imagine, a little bit bigger than an eyedropper. But this kit can, and then on the left is the paper devices, which are about that big, and you can carry them around. So you can do these assays that typically would be limited to the lab, but now you can do it in a portable setting, where in this case, it was meant to be more human health related. So it was meant to be marker for sepsis, and you could see the result just visually. You see on the right, on the control zones, you'll see like a clear white area, and if the target is present, so in this case it's C-reactive protein, it's a marker in blood, you'll see a dark color develop, and you'll be able to tell whether it's positive or not. And we showed that using this kit versus using lab equipment like micropipettes, we were getting very similar results. So that's what my background has been. It's like trying to make these assays and tests that are really limited to the lab right now, but trying to bring them make them portable, make them easier to use, make them more widely available.
And so what's the reaction time for a sensor like that? Is it minutes?
It varies depending on what you're trying to detect and which techniques you're using. And in this case, we're trying to go somewhere between 30 to 60 minutes to get a response that is visually clear, that you can see from the naked eye. And that actually brings up a good point, because right now there are lab-based tests, right? So you could take the sample, ship it off to the lab, wait for the results, and then act accordingly. But that will take at least a day, if not several days to weeks, as you've seen with the COVID-19 pandemic, right? Like you try to get a test early on, it takes a long time to get the results back. And that's what we're trying to reduce that time, so you have the information right there.
Okay, and I guess it doesn't require a sterile lab. I'm guessing Aaron's livestock barns aren't necessarily a sterile lab. Like you get a sample of, you get them safely into the chute, and you can swab their nose or something and put a drop on a piece of paper. Like what turnaround time would be good for you, Aaron, there on the farm? Like what would, is finding out in 30 minutes? Is that good enough?
Well, immediate's great, but you got to compare it to what Mohit mentioned today. I mean, how this normally works is I'd have an outbreak of cattle that I've tried a lot of different drugs and I can't find one that seems to consistently work. And so I'll take one of the ones that died and autopsy them and take, parts of the lung and other pieces and send them off to the lab. And that just the next day shipping is like $160 to send this thing off. And then it gets there and they're going to run some initial tests on it, but then they're going to try to speciate what's there, and they're going to try to grow the different versions of things on plates and put antibiotics on the plates and figure out which antibiotics it was actually going to respond to. And so I'll get that response like 10 days later. But by then, all the ones that were going to die had already died. And so usually I don't do it that often, just because it just comes back and tells me what I already gave them probably should have worked. And, they had BRD. Well, thank you. $400 later. And I'm none the wiser, but you feel better when you send the sample off. So, the best thing is one where while the calf's in the chute, I can do it, but I'm doing this every day. And what I'm really wanting is this sort of data feed. I need this stream of what have I normally been seeing, because there can be some false positives, there can be some false negatives. I need to know, is this calf one that's going to be in that like 15% that's not going to respond to a certain treatment because he happens to have different stuff, right? And so to me, the ability, even within 15 minutes, half an hour while we're there working the group, we could just be putting a sample into a device and then getting answers back as to what we're seeing today. That's just miles better than after they're all dead and then we figure out what they had. So I'm excited about it, because that one calf out of 100 that I happen to autopsy is not a statistically significant sample. He may have just had something completely different than everybody else, and that's why he died first. And then I've got a completely wrong knowledge about what he was even going on. So
yeah, very good point about this data set that's growing, like a stream of information. Yeah, really pertinent. So I'm sort of kind of thinking back of, well, it wasn't that long ago that you two met, and the whole problem surfaced to Mohit, and Aaron's probably laid out his dream very nicely on a piece of paper, like, please deliver this, and last week would have been great. So how easy is it for a person like you, Dr. Verma, to generate a solution like this?
Well, on paper, it's always easy, but once you start working on it, Aaron's always like, wow, well, that's taking way longer than I thought it would. So no, so I mean, so once we got this idea, okay, we should try to do different types of bacteria and viruses, we looked into different techniques that could be used, and then we came up with this method, which is a molecular diagnostic method. So it's based on detecting the DNA or RNA, so the nucleic acids from the bacteria or viruses. If you've heard of the PCR, it's very similar to PCR, but it's called loop-mediated isothermal amplification. And
now I know exactly what you're talking about.
Short is LAMP. So really what it is it's a technique that can detect different types of nucleic acids and in the presence of a single temperature. So in PCR, you often need to take the temperature up, bring it down, do this very precisely. In this technique, you can just keep it at one temperature. It will amplify the nucleic acid that you're targeting. So let's say we wanted to look at specific bacteria, it'll target that. And then if that is present, it will cause a color change. So you'll be able to see the color change visually and know whether it's positive or negative, just like we did. So we started working on this technique and then looking at the different types of bacteria, looking at the different types of viruses, and we've been doing that for a while. I mean, obviously, there's been a lot of challenges, but we're actually pretty close where we can go out and answer that question, Dennis, that you asked earlier, will it work in the farm? So we actually have a crude setup, but we can take it out in Aaron's farm, collect some samples right there, try to get the results right there. And it's working fairly well, especially the sterility part. Our controls are staying negative as they should. So that's working out pretty well.
And it has been fun for me to see graduate students that have never been on a farm, come out in the winter and walk through the pens with me. That's been the best part of this entire project for me. Yeah. Those guys have been really great.
So I'm kind of jumping ahead, but I have to throw it out here now because it just came to mind. Before we let students come back to Purdue campus, we had them tested to make sure that we're reasonably confident that they're not bringing COVID back. So I could certainly see one application of this would be particularly feeder cattle. You're sending them to an auction somewhere like, oh, you got to have your BRD free test out of Purdue before you can bring your cattle to this auction. And extrapolating, of course, I'm thinking it probably isn't too far into the future. We have lots of tests that are like this for common things that are spread in the community. I mean, it's interesting that we're talking about it in the COVID-19 era, but like you're saying, Aaron, they catch this from each other. And once you have an outbreak, well, then, that sort of a thing. So anyway, back to, so tell us a little bit more about how this one works. How sensitive is it, Mohit? How confident are you in results, that sort of thing?
Yeah, so I'll actually jump off of what you were just mentioning, which is because it is based on nucleic acids, which means DNA and RNA, which build up everything, all life really, you can target different diseases by changing the target sequence. So right now we're doing it for BRD bacteria and viruses, but as the pandemic broke out last year, we actually jumped right in and developed a test for COVID-19 because we had this technology that was under development already. And as the pandemic was breaking out, we had the right partners to try to develop a test for COVID-19 itself. And we started last March, just as everyone else around us was shutting down. We started working on this, and here's what we ended up with. So over a period of a few months, the way you want to think about this is initially, a lot of the tests were just not available for COVID-19. And what we wanted to do was simplify the testing by using saliva samples, so you don't have to take those deep nasopharyngeal swabs. And that's what we developed over here. And what I'll point out is, if you can see my cursor, can you see my cursor, Dennis? If it's
a little bit.
Okay, I can also just do this. All right. So it's really like a kit, just like I showed with the paper device, but this is more like a prototype. So what happens here is there's a cartridge over here, which you see shown in this green handle. And then there's this heater box, which is shown in gray. And what you're doing is once you collect the saliva sample, you are taking some of that saliva and adding it to this cartridge. The cartridge has all the reagents that are necessary to do the reaction. So it has enzymes, it has primers, it has these dyes that will change color. And once you add the sample, you put the cartridge into this heater. And then in this case, it was 60 minutes because saliva was taking a while. After 60 minutes, you get a color change and you compare it against a color chart. So you can see whether it's negative or positive. In this case, red would indicate negative, and that's what the control should look like. And if there's virus present, it will turn yellow, and you would be able to see that visually. So that's how the test works in the case of COVID. Now, as I was saying, because it's nucleic acids, you can change it up and then apply it for BRD, and that's what we're doing right now. We're essentially taking a new target, building upon this to try to simplify the whole testing process.
Okay. So which do you find easier, COVID-19 or bovine respiratory diseases?
Actually, COVID-19 was the easier platform because you only had this one virus that you had to target. Whereas in the case of BRD, you actually have so many different bacteria that you can detect, that you need to detect so many different viruses, and also the antibiotic resistance is a big problem. So it's actually more challenging because you need a multiplex test to be effective, and that's why it will take a little bit longer, but we're working on it.
Okay, good. So I guess first you need sort of all the genomes of the diseases or whatever.
Yeah, so exactly. A lot of these genomes are available, but a lot of the things that are not known is what is the prevalence. So Aaron was mentioning that a lot of these are present, like just commensal organisms, which means they're present all the time, but in disease, they go higher or they infect somehow. So there's not a good survey yet of what is that difference? Like, is it 2 times higher, 10 times higher? So I'm working with collaborators on campus to try to determine that, like, what are the levels that are present normally? What are the levels in disease cattle? And that's part of the sensor work that will guide, okay, this is when you should say they're healthy. This is when you should say they're sick.
And I'll say that amazed me that we didn't know that information. We looked, it wasn't out there. I mean, BRD is the thing that you deal with in cattle. That's the problem in beef cattle. Everyone has it. I don't even know the numbers off the top of my head, but it's insane how much money and time and so on is lost to that disease. It's the thing. And to think that we couldn't find, well, what is the concentration of bugs in the nose of a sick calf? Like, that seems like we should have known that 40 years ago. And so it's been nice that we have a reason to do that now and learn that. But the downside to that, though, for me was that I said there wasn't very much information that you did know when the calf was at the chute. And the one thing you could have was their temperature. You would take their temperature, and if it was high, you assume they were sick. And sadly, this study has completely shot that in the foot for me. I've learned that the temperature is largely irrelevant because in the past, you never pulled healthy calves and took their temperature. And so you just assumed that when you got a high temperature that you did a great job picking that calf out of that pen because he was sick. But we've been pulling healthy calves to try to figure out this relationship. And most of the healthy calves had temps as high or higher than the ones who are showing all these symptoms. And you would see nothing wrong with this calf. It's fat, it's jumping around, it's happy, there's nothing wrong with it. And so, it just has taught me to put a little less faith in the temperature that we would take over time. And then that just made me need this device all the more. Because now not even the one thing I thought was the thing you could count on, a fever, you can't even count on that. So
this is really, I think it's really timely that you're now studying and talking about this in the current year. Also, because in the case of COVID, we talk a lot about asymptomatic people. I'm sure there could be asymptomatic BRD carriers in the cattle, but it is very interesting that Mohit, it sounds like your test is showing like positive, well, I'm going to use the wrong term here, correctly showing these are not carrying, they're not only do they not have it, they're not displaying symptoms, they also are not carrying it. Boy, if we could have that sort of a test for us, that would be super. Yeah. So do you have any, you're an engineer. I think this is a wonderful intersection of sort of sensor engineering, biology, physiology, practical farming, livestock farming in this case. It really is a wonderful intersection of topics. But I feel like, I'm going to need to see a chart or something like how accurate is this thing.
Yeah. And I will actually point out to our most recent results. So what we've done is just taken looking at different pathogens. We took the top 3 bacteria to start off and started looking at, can we design the assay for these bacteria? So we're looking at Pasteurella multicida, Mannheimia hemolytica, and Histophilus somni. And what you're seeing over here on the left is on the x-axis, so these ones over here are different isolates of the bacteria. So PM stands for Pasteurella multicida, MH is Mannheimia, and HS is Histophilus. And then these are just controls. The heat map shows the intensity that we're seeing in terms of response. So the darker it is, the more of response you're seeing. And then on the y-axis over here are different types of primers that we use to detect these bacteria. So an ideal result would just be a diagonal like these diagonal blocks shown in red border, it should be showing you a result. And that actually looks very good. So most of your red blocks are filled, and there's very little background noise or background false positives over here. And when you convert this data to something called a ROC curve, or basically a receiver operator characteristics curve, you can determine how good is your assay. So you can look at your false positives versus true positive rates. And we see that it's really good in terms of detecting the species that you want to detect and not detecting the ones that you don't. So it has greater than 95% specificity and sensitivity. And right now, this is all done in the lab, right? So now the next step really is how well does this work in the feedlot, and do these performance characteristics carry over moving on to the feedlot? And that's where we're going next. So far the assay looks really good. Now we're going to try to see how well that maintains clinically.
Awesome. And if it was only available for 5 cents a test, this will be wonderful, wouldn't it, Aaron?
Yeah, that would be great. I would take, somewhere south of the 400 I have to spend now, and even get the answer sooner would be nice. But yeah, in Histopolis somnus, you had that listed there. That reminded me, when I first had some of that a few years ago, or I thought I had some, I was looking up, some more information about it, find some research papers, read it so I could figure out, how to identify it sooner. And it stuck with me to this day. There was a line in one of those that said, oftentimes with Histopolis somnus, the first symptom is death. I thought, that's a tough one. Like, how am I going to catch that calf?
Yeah.
So that, probably the first symptom is the presence of the bacteria if we had to test for it, which would be a significant improvement over death. So.
Yeah. Well, maybe we're going to have to give you, Mohit, what's the nickname? I guess that's what I was thinking of. We'll call you Dr. Doolittle, because in this way, you're essentially teaching these cattle to talk. Like, this is what I have today. So they're able to tell the veterinarian or the farmer, how to treat it, that sort of thing. But I guess in order to get to that point, it's got to be commercially available. So where can I buy one of these? Buy a kit, a stack of kits, a pallet of kits. What's the process from here?
Yeah. So of course that's the goal, right? In the end, I want to give Aaron a test that he can go and use it by himself, right? And that's why we're looking at taking this technology and trying to commercialize it. We have the IP filed on the primer side and the actual technology and working. And I just founded a startup company last year, Krishi, and we participated in the Purdue Ventures accelerator competition, and we were successful. So we have investment from them. So that's really helping us think about, okay, now how do we turn this into a product? What do we need to do? How does it fit into the workflow of the customer? Who's going to be actually buying this and using this? And so that process has started. Hopefully within the next couple of years, we'll have some prototypes that can actually go out and test more easily, I would say, more easily, so that they're more user-friendly. So that's the goal through a startup company.
Cool. I think it is, this is kind of a unique partnership that you two have going. And I know that it's bigger than that. You have a larger group working on it. But I suppose it did help that Aaron, although he is a part-time Purdue employee in our Open Ag Technology and Systems Center, that sort of lends him to be pretty naturally inclined toward research and that sort of a thing. So Aaron, how does it make you feel to propel this work of that you guys are working on?
I told Mohit when this started that this this would be the most important thing. If it worked, be the most important thing I ever helped make go. But it's really weird for me because normally I'm the guy that has to deliver. And in this one, I'm just the guy with the need. I cheer on Mohit's team and say, you guys are doing great. It's been so strange here to, only be the domain expert and not have to also bring the solution here. So thankfully they've been doing a great job. Because I'll be honest, Mohit, I don't know if I've ever told you, but I was a little skeptical the first time. You were like, we can just put gold nanoparticles in a little solution, it'll turn purple. And I was like, that's really cool. I didn't tell anybody that was coming yet, though. I hedged my bets, but actually now, you know what? It's happening. And so that has been the most exciting thing for me to see, that that this actually has a shot at making a significant difference in this industry. So I'm excited.
Yeah, really cool. And also this is a good reminder of the role of land grant institutions. And Aaron, in this instance, as the need provider, but the direct connection of what the need is and the aptitude, the skills, the laboratories, etc., of the person who can generate the solution to the need. Like, it takes both. And so, yeah, necessity is the mother of invention, and the land grant institution is often the propeller of those solutions. Yeah. So anyway, this is just a great topic, and great work all around. So maybe before we head to chat there and see what others have posed as questions, any other sort of just thoughts about the process along the way from either of you two, and where you're going from here, that sort of thing? Certainly.
I think I'll actually build up a little bit on your final comment, Dennis, there, which is that the two of us are here, but it's actually a much bigger team behind this. Sure. There's people from animal sciences, veterinary medicine, all over the campus, really, that are helping us. And we've been successful because of them, actually. So I didn't know much about agriculture, even like when I came here. I knew biosensors, but I didn't know much about agriculture. But after I got here, working with Aaron, working with these partners and collaborators, that's why really we've been able to make progress, and we've been successful securing funding externally and getting some attention and really make a dent at this problem, I think. So really, shout out goes to everyone that's part of the team.
Yeah, and I'd add too that the team has been able to put together, I believe, a 100% success funding rate on this project, right? We have not lost a single proposal that was written. They all got funded, which is I've never seen. So I think that speaks to the prevalence of the problem, and probably the way that the team overall is able to, say concisely and clearly what exactly we're trying to do. So that has been great. I mean, for me, the most exciting thing for me, maybe it's the data science part of me, is statistical significance. This is the only way it's ever going to happen. And I sometimes describe farming as engineering without the formula book, right? Like, you're going to take the test, but you don't have the formula book. You have to make a decision today about something, right? The calf is sick. We are going to do something, and you have little information about it, like so little. And you know that there's this big statistical mystery somewhere behind the scenes that you just wish you could figure out. And so to me, it's so exciting to think that we could finally get something that I could gain that statistical significance. I could know when I have outliers. I could know when I have things that are under the curve, and to know that sort of in the moment. I mean, that's sort of the holy grail of all data problems, right? So
yeah, awesome. I saw a little bit of chatter in the chat just about Fitbits, that sort of thing. I'm just curious, do you have any in your beef business there, Aaron, or is it only dairy cows and highfalutin race horses that have those things?
That is a good question. Yeah, the problem with beef cows is there's not a ton of margin per animal. I mean, there's a lot more than like a chicken, but it's still not enough to justify a very expensive device to try to track their activity. So there are some companies out there, like Caithness, I think is the name of one of them, that they'll try to use video to track activity and things like that of animals. And I think that's useful. The stuff you have to put on every animal, though, is I'm not sure that it'll ever make it into the beef industry as a whole, just because there's just not as much margin. In the dairy industry, there's huge margin per animal. Well, if I were a dairy farmer today feeding $6 corn, I wouldn't think I know there's not that much margin. $16 milk and $6 corn don't go together. Yeah. But yeah, so we don't have them on the beef animals just because there's not enough margin. But I think that kind of a stuff would be extremely useful, especially if you had some of the results from these sensors, right? That we could start looking at what kinds of things do we see here in conjunction with these other signals, right? Activity of a pen, the feed rate of the pen, those kinds of things. So they are out there. And then funny story, the first proposal I ever wrote at Purdue, right, fresh out of college, was actually one to create a little biosensor that you would put in the cow's stomachs that we could read externally to get their temperatures and to get the pH of their stomach and stuff. And that one did not get funded, of course, but it would have been dairy only as well. But.
Yeah, cool. And even though you might have that other, like you say, it's really a sensor fusion situation, because even if you detected number 22 is less active than it had been for the previous week, you still don't know what they have. And therein lies the value of a specific sensor. This is what it has, and this is the right treatment for what it has. Yeah, yeah. And we haven't figured that out for humans either. If I talked about treatments, I'd be making a political statement, which I don't want to do right now. Any questions from our participants out there? Feel free to unmute and pose a question to either Aaron or Mohit. Guys, I really appreciate the conversation today. This is just motivational in several different regards, not just the BRD part. Yeah. Anybody with a question, feel free to just unmute and introduce yourself. That would be great.
Yeah, and I can add too, Dennis, that the external ability to sort of monitor that beef herd and get some of those signals. What would be really nice would be to, when I go walk into the pen to look for sick cattle, I've already got a list with me that says these are the ones that we think may be having problems. You should pay attention to those. And I actually remember I talked to some guys over in mechanical engineering, but we never got the funding for it. They were looking at making some augmented reality glasses. And I said, man, the thing I need for that is when I go walk in the pen, it needs to highlight the cow that I'm supposed to go look for, right? If you had the list of the ones that you knew you needed, and then I could look around and they were all shining red, man, that would be cool. Maybe that'll be next, Mohit. Can you pull that off too? Do they make a sensor for that? You've been a miracle worker.
Maybe one day.
All right. Any questions out there? Or do either of you two have a question you'd like to ask people that are attending today? That's fair too. They're here. They're participants in all of this.
I will open it up. Yeah, if no one has, I'll open to the participants. Where else can you see this technology going, right? Because I'll even seed it because you've already seen COVID-19, you've seen BRD. We're doing some food safety work. We've heard a lot about pigs. So pigs have a lot of diseases which have similar problems where if you could find out right there, it would be very useful for management practices. Other ideas, where else do you see it going? Put it in chat. There's one about crop disease identification. Yeah.
Yeah.
It is a good idea because that's the nature of how the scouting of a cornfield isn't that much different than scouting a lettuce patch, right? So it would be, even though raising produce is completely different than raising row crops, that same model would probably work really well. That's a good idea.
Yeah. Certainly, you mentioned the food safety work that you're doing. That is super valuable to be able to, even if there is a slight delay, to at least know which lots, which batches, etc., ahead of consumption. Anyway, that always pays off. Yeah.
And there's a different idea there too, which is more from the risk management perspective. Like when you're planting as well, you want to know, is this a safe field or not, right? So are there potential for contamination and so on. So we're doing some work, and actually Aaron initiated this too, but we're doing some work with the Center for Produce Safety where we're trying to determine, are there areas which are more likely to be contaminated by produce, by fecal contaminants, or are there ones that are safer? We're trying to find that out. Same technology, different targets.
Cool. Well, I think we'll go ahead and wrap it up if there are no questions from the audience. But Dr. Verma, Mr. Ault, I really do appreciate your participation today. This is quite stellar work, and we're glad to hear of it and sort of promote it through the webinar series. So just a reminder to all, we'll be back again next week and 3 more after that. certainly welcome you back. This will be the upcoming topics. John Scott, I believe, next week. Raghu Ragamathan coming on April 29. I've got an ag economist, Nicole Widmeyer, on the May 6th, and John Evans from ABE and maybe Dave Capillari with him on May 13th. So thank you all for participating. Hope you really enjoyed today's presentation and discussion. Have a great, safe, and productive and enjoyable spring.
Thank you for having us here, Dennis.
Yeah, thank you.